G1, what happens at G1? Initiator proteins, excellent. So we have regulation of genes, transcription. We translate regulator proteins, initiator proteins. Those turn on, you find out points of origin, open up your replication bubble, so forth. Then we come together, we snap, we finish meiosis 1. And spermagenesis, we start with a stem cell, we undergo meiotic division. And then we go through meiosis, and you create how many daughter cells? 4, haploid, diploid. Haploid. Okay? Then they undergo development in the testes. We know all the locations and so forth. Fertilization happens. Okay? At fertilization, sperm meets egg. What takes place? Calcium wave, right? Calcium wave takes place so you don't get polyspermy. From there, the receptors all fall off. Jellycoat expands, the lab you guys did online. Okay? And then what completes? Meiosis 2.
Meiosis 2.
Is complete. Okay, we have a lot of refreshing to do.
Yes.
Right? First question on the next exam is going to be, start at meiosis, go to neurulation. Okay? So make sure you know this. All right. Then we undergo what? Cleavage. Excellent. We create a blastula. Blastula is what cell types? Totipotent. Toti means they can become anything. All right? They're going to replicate. We're going to make a whole bunch of cells. Those cells are all connected together. Then we're going to go or change. What is that change? Gastrulation. Excellent. What happens during gastrulation? Formation of ectoderm, endoderm, mesoderm. What does it actually mean? What has happened? They change the type, go from toti to pluri, but what actually happened? We haven't formed neural tube, that's neurulation. We haven't turned them into anything, that happens next. What actually happens?
Formation of receptors.
Excellent, formation of receptors. The receptors change. This is going to hurt your brain. Tell me, what receptor is found on ectoderm? Talk to each
Where am I?
What is the receptor found on ectoderm?
TGFB.
TGFB. If noggin is detected, it turns into neural tissue. Otherwise, BMP4 is there blocking it, turning it into skin. This is during neurulation. We go from pluripotent to determined. And then the next thing we go to differentiated. In ectoderm, we create a neural tissue and we start creating segmentation. Runs down through the body plan as Henson's node traverses the embryo. We form little repeating segments created by Hox genes. What are those repeating segments called? Somites. What do somites turn into? Spine. Okay? So if we have a defect in that, like spina bifida, which was a very hard question on the exam apparently, which I forgot to bring the exams, I'll bring them on Wednesday.
I know I did.
Repeating segments that will have effects. Now we undergo the same processes occur in mesoderm and in endoderm. which is going to create our different organs and organ structures. That is where we stand off today. Okay? Where we're going today is we're going to do organ structures. And we're going to start with nerves, because we started with ectoderm and we started with neurulation, so we're going to start with the formation of nerves and action potentials. That's what we're going to do tomorrow on Wednesday. Okay? Then we're going to start going into mesoderm. Mesoderm, we're going to start because nerves start functioning on muscles. Muscles are mesoderm. Okay? So we're going to get how muscles work. And then we're going to start understanding how specific types of muscles work, like blood vesicles. Aorta. Aorta is a muscle. Okay? Your arteries are muscles. Your heart is a muscle. Right? All of these components, that's your mesoderm. And then we're going to touch a little bit on the endoderm with the gastric system. That is 223. Okay? And then we'll sprinkle a little bit of plants at the end. Yeah.
So do we have lab on Thursday?
You do not have lab this week.
Oh, no.
Okay? You do have lab. It's online. It is hard. You're going to do all of 231 across the hallway this week. It's anatomy. Okay? Anatomy is simple. Memorize it. I'm not gonna cover it. We're not gonna do anything. You are gonna memorize it. Create flashcards. Look at the key terms. Just memorize. That's all anatomy is. If I say what a carpal is, you'll need to know what a carpal is. Okay? You got this. So no lab this week, or even next week actually. We're gonna start planning cures. We're gonna talk about cures here in a minute. The first lab that we have is week 3. That is human health. Human health is a long lab. It's going to take the whole time, maybe even a little bit longer. Human health, we are going to cover everything from EKGs to urinalysis, blood work, everything in lab as if you were at taking a physiological thing. Then we're going to do some dissections and we have a few labs. So we only have like 4 or 5 labs actually in person this quarter, but we have your cure. So, I saw that some of you are starting to think about cures, so let's talk about cures. What makes a good experiment? So let's go back all the way to 221, that first week.
A good hypothesis.
Good hypothesis. Tell me what is a good experiment. So, recap, talk to each other for a second, tell each other, what's your ideas, think about this for a minute, or spend 5 minutes on this, and what's a reasonable experiment?
I put my name on the cure sign-in, but I don't know what I'm going to do just yet.
Yeah, I think I'm going to clone a plant or that's the idea, and figure out if when you clone it, how does it if you put light or if you don't put light, which one grows faster? Something like that, or maybe I could do colors.
Something basic,
Yeah.
you could say.
What about you?
I was thinking developmental toxology, so developmental embryos affected by different toxins that would be in there.
What we did, so we're going to talk about that in a minute.
Would that be kind of like what we did with the embryos? Like you put, like we put alcohol in cases, but in this case it would be
Just
That's interesting.
It's fun time.
We'll talk about that.
That sounds interesting. Have you thought about something?
I was thinking about water pollution, like from different places. You're gonna test it and compare. And after that was like original water source, and like when the water system was renewable, what was the difference between the result gonna be different?
In water sources?
Yeah, like from home, like from you and mine, it's gonna be different. So try to figure out the reason, like check source where we got this water, like original one from where it's taken, from which water, like.
You know who's also doing that?
I don't know, somebody else doing it.
Yeah, I think she's next to Bodo. Maybe you should get together. I thought about when she put it there, I was like, maybe I could do it. I always was interested in that too. And you haven't thought about what? How many people did he say could do it? Like only 2? 2 to 4?
He said 2 to
Oh,
4.
good. Maybe better if I could, I don't know, join somebody, like if you have a...
I was thinking of joining somebody else.
Oh, but wait, we have to be in the same lab, and we're not in the
We
same
have to
lab.
be in the same lab section.
He said it's better. The same
It's better,
time.
yeah.
I think you can talk with him and change
Huh?
I think you can talk with him and change your laptop.
Yeah, that's the thing. I don't think I want
Or
to
you
change
can,
it.
or you could do it between labs.
Yeah.
Except like one just got out, one's just going in.
Yeah, because I think we just do it on the weekends or something, or in our
Yeah,
spare
it's
time,
probably-
or we could just be like, you do this, I'll do that, and then we come up. I don't know. Let's ask him. So, what would make a good experiment? He said. Something, because I asked him about something regarding chromosomes, and he's like, no, but that's already done. Well, I mean, I think everything's, or not everything, but everything that we're thinking about has already been done, right?
Yeah,
Exactly.
so experiment is fun. You can actually test it, so you can what is testing the machine.
You can test.
Yeah.
Something measurable, repeatable.
That you can measure.
What makes a good experiment? How long do you have to conduct this experiment? About 6-7 weeks. How fast is this quarter going to move? Really fast. So we're going to come back to your senioritis thing here for a second. Our running start students in here are going to struggle immensely this quarter. And the reason why isn't because things are going to get harder. It's because you've been admitted to university, you're graduating, and you've been in school a really long time, so you feel free. You haven't graduated yet. All right? Whatever happens after June 10th, I'm cool with. We make it to June 10th, okay? We struggle through, we power through. So I do recognize that. But how quickly is our CURE project going to go? Really fast. And are we dedicating a lot of time theoretically to this from lab?
No.
Yes. So do I expect a high caliber project? Yes. You guys have seen the posters out in the hallways. You also know that we win every year, first, second, and third. All right?
No
we have
pressure.
high standards in here. So let's talk about what makes a good experiment. Anybody know, because we're starting to think about universities, what makes a good PhD? This is not a PhD we're doing here, but what makes a good PhD? One that you finish. Understood?
Yep.
So what makes a good experiment?
One that you
One
can.
that you-
One that you finish. Excellent. Pick it on you. Because you're the only one who turned something in already. You did. You turned in a document of what your experiments, your ideas were. So I'm going to pick on you guys because you guys have done good work. But it's not reasonable.
Oh.
It's a huge, grandiose project with multiple different facets in there. They wanted to go do and measure a whole bunch of things outside in the environment. And then they wanted to take some of those and measure and manipulate and do experiments in lab.
Time.
Wrote. The pH and all those things and the environment. Yeah, keep going with it. Yeah, that was it. It's been a long time. Just leave it. It's fine. Each one of those individually is reasonable. Together, not reasonable. We don't have enough time. So if you want to go out and you want to measure the salinity and the pHs and the chemistry of the different lakes, cool. That sounds awesome. Sounds wonderful. You want to go look at microplastics in the environment, cool. Sounds awesome. You want to go then look at the microplastics in the environment and then manipulate things on clams, That's a PhD project. Because that's too many components together. Yes, really cool idea. Love the idea. It's amazing. Great thought process. Can you do that in 6 weeks? Probably not. So the experiment that you're going to do is one that you're going to finish in 6 weeks. Realistically, if you are doing measurements out in the field, it's 1, maybe 2 times out in the field. Maybe 3. Okay? I know as an ecologist mindset, you want to be out in the world, out doing things, out in the forest, right? You go actually talk with an ecologist. How often are they out in the field? Not a lot.
Once or twice.
Once or twice a year, a season. They're spending so much time getting everything so they understand how they're doing it, what they're doing. That's what you're going to do. So for the first 3 weeks in here, you're probably not doing any experiments. The first 3 weeks is you are studying, you're learning what is known, right? So, I'm going to pick on you, sorry.
Who's next?
You just mentioned, what if we take and we dissolve something to understand the pH of its reaction? Like of a pill, right? Cool, but you're not understanding how it's uptake into the body, right? And we're not manipulating anything on animals, okay? Insects, cool, sure, fine, whatever. Vertebrates, no, nothing with vertebrates. But if I were to Google, how does pH affect this pill, what do you think happens? Pops up. The Google AI tells you exactly what happens. All right? So is that a reasonable experiment?
No.
No.
No. Right? A reasonable experiment then in this would be, okay, let's manufacture a pill. Let's do it. That's a reasonable experiment. I'm sure Samita would love to do that. In fact, two years ago, we had a student manufacture a protein, did protein synthesis. Her idea was to make a trimer. She made it a dimer. That was really awesome. It got her into graduate school. That project, she is now in graduate school for that project. So, like, if you want to make a drug, sure, cool. What do you guys do in chemistry now? You guys make aspirin? What do you guys make? You guys make aspirin.
I
No,
make
not
Bochem.
no chem. This is probably in Gen Chem. Did you guys make an aspirin?
No, we didn't.
Tylenol?
We didn't
We
make
didn't do
them.
any chemical synthesis.
We just boil water.
Do you know what we used to make when I did chemistry? Meth! We used to make meth in class. That's the cool shit. You don't allow that anymore. Now they make you bake Tylenol.
Boring.
It's so much cleaner. Ask Samita what you used to be able to do in life, and you used to like play with mercury and move it around and uranium. You pull it out and throw it. Sipping. You guys have bowl pipettes. That shit's cool. We used to have to suck with our mouth up. And if you inhaled the benzene, that really sucked. Okay? There's a reason why life expectancies are going up. Okay, let's just start with that. All right. So if you want to make a drug, let's make a drug. If you want to experiment, you want to do something in the lab, let's do it. If you want to do manipulation on plants, let's do it. If you want to go out and you want to measure butterflies, go out and do it. So whatever something is, let's reasonably narrow it down, tailor it. How many variables are we going to manipulate? Ideally 1, right? Now, in the real world, that's not usually true, especially if it's a survey. Surveys are you just exploring, right? Yeah. So, any questions? Yeah.
So when you say survey, is that an experiment?
Surveys can be an experiment, right? So, the other aspect of this is if you want to be an ecologist, I expect you to be doing ecology things. All right? Use this as your stepping stone into the next future. All right? If you are somebody who really is interested in plant biology, I know we have a couple of them. Let's do something with plants. Let's do manipulation. You have a greenhouse. Let's actually use it. If you are really interested in, you remember that experiment with termite behavior and them following needs. If you are that drew your mind and you're really interested in, behavioral something, let's design a different experiment using something similar, right? Use what you know, expand out. Okay? Questions? All right. This week, I want something from you guys. So I want to know who's doing what, where, what your thought processes are. Could be a 3-minute conversation, phone call, something. I want to touch it. On Canvas, there's a whole bunch of stuff. Get that done. Due this Sunday, project details. So don't wait to do this. I want to actually talk to you in the week at some point. And then we need by April 20th is our rough outline. Full. What's going to be happening? What you need. What you need is a very important thing. If you tell me, hey, so you want to synthesize a drug, right? But we need 2 compounds. Well, you have to give me lead up to be able to get something, okay? So all of a sudden, if you turn in something April 20th or April 28th when we finalize stuff, and it's a 6-week back order on something, that really sucks. So get to me pretty quickly with that. Cost. I have never really turned down a student on cost. So we do have funds available to us. There's a decent amount of money. With that said, like thousands of dollars probably will laugh at you, but none of your experiments costing thousands of dollars. Actually, it's not entirely true. A couple of years ago, we had an experiment that ran about $20,000. It was a really cool experiment. Can't do it this year because I don't have any grad students doing stuff right now. But they worked in my lab and they did isotopic analysis. It was about $20,000. So, yeah. So with that said, let me know. Come up with what you specifically need. That's very important. If you also are needing to do stuff in the field, I have some rules on field work. We never go alone, ever, by ourselves, okay? If you are doing stuff on mud flats, I have to be with you, or a faculty member has to be with you. The most dangerous thing you guys could be doing in here is stuff on mud flats, okay? I have had to have safe students before, and I've had to have myself safe before on mud flats. Mud flats are very, very dangerous. So, like, if you want to do an eelgrass survey, eelgrass surveys are really fun. You get to go out in the intertidal, you get to measure stuff. It's actually really cool. And we can work with like the Friday Harbor Laboratories. We can send you up to Friday Harbor for a weekend beyond our Friday Harbor trip. But mud flats, we have to be careful, okay? If we are going out in the middle of nowhere, we need to know where people are at, so forth. Make sure that if there's a permit involved, like we actually do the permitting and not get arrested. I had a student who almost got arrested one quarter because they tell me they're in one location, all of a sudden they're in the middle of nowhere and they're doing a tree survey and they're on somebody else's private property. So we're not going to do that, okay? Okay. If you want to turn this into our honors, let me know if you're in the honors program. But all right. Any other questions? Thoughts? Yeah.
Can I see that you sent an Excel out to write what you're doing on there, and I think it was in the last section's Canvas.
Yes.
Can you resend it?
Yeah, it should be in this document too, on there. But yeah, so that's mostly for you guys to communicate at this point and kind of self-organize. I do not care who you work with. I personally don't care what project you do. I want you to do something that's meaningful to you.
Thank you.
All right, other things. I think our field trip to the zoo is a good go for this Friday. We've had a lot of awfulness working with the college on bands. They changed the rules this quarter. And so now we think we found loopholes in the rules. So I love loopholes, like non-love loopholes. So the field trip dates are set. They're in the schedule. May 3rd, I think is what it was, for the San Juan Islands. Please sign up today for that. if you think you will probably be going. If I get to a certain number of people, I can get a bus with a driver. You kind of cool. So let's talk
Sign up,
about the
sign
San
up.
Juan Island trip
Right.
for just a second.
Are you coming?
We will leave here
Someone-
about 7 a.m.
Island?
Friday Harbor?
Yeah.
Go up to Anacortes and get on the ferry. From there, we go to Limekelm State Park on Friday Harbor. Then we're going to go to
Gonna
meet
be
up Friday
fun.
Harbor Laboratories. Where you'll do a tour. One more. They have their open house that day. And so we'll hang out there. We'll have lunch there, and then we'll go to Cattle Point, where we'll do kind of a couple field surveys. The tides are going to be beautiful. That's a -2 tide, so we'll be able to do some tide pooling. There's a couple little hikes.
We should
The
be able
hikes
to
are
go with-
very marginal. They're like quarter, half mile.
Would I be able to go?
It's a fun day. We'll get back by about 9 to 10 p.m., so it is a long day. So yes, please sign up. If we hit a certain number of people, we will do a bus.
Are you going to see me?
Sign
No.
up. It's gonna be fun. It's a lot of fun.
Really? Darn.
Let's get started on PCR. What?
Molecule. It is in as much detail as you can.
It's most likely to recruit chemistry.
Black is given as an example.
PCR.
But yeah, you need to look up how to actually chemosynthesis. Yeah, you're not. You might need an
It's like right up your alley.
And your alley. Yeah.
Okay, bet.
It's not a very well, you're not going to be also not synthesizing something. But it's only valid as a treatment too. You're like, hey, I'm going to be a pharmacy major and biochemistry. I'm not going to send you. But if you're an ecology,
Yeah, that's it. Here's the dry.
that's not going to help you.
It doesn't matter if it works or not. What matters is self-testing. I'll take
So
it.
that's something you'll need to think about. And two is the lab technician. And she is a chemist. Right, so that's something to
I am going to talk to him about it. I do want to try my
It's probably See, I swear to God, you guys do synthesize. Over there.
Oh, did I?
Big word. Small raise.
Things like that require huge setups. Usually it's just going to be something very simple aromatic compound.
I knew it was like clean.
PCR. Oh. Well,
What is the project over there again? Oh, yeah, definitely. Oh, yeah, because you guys are like monitoring.
It was just aerial photographs that we're analyzing. I couldn't see.
So you go out and actually do a field survey, field graph.
elongation.
Yeah, I say go out and do it.
It's one of them that's
Okay, I'm talking about original DNA.
so
Okay, let's start right here. This is DNA, right? All of it?
Those are like triphosphates. Those are like, this is ATP, this is GTP, this is TTP, and that's CTP.
Oh.
I remember seeing something
Wait,
like that.
T what? TTP?
Yeah.
And they're triphosphates.
Yeah, triphosphates, yeah, essentially. I'm trying to figure out what the green and blue are. It's just,
What do you have for green and blue? Nothing. What does Elia, what do you have for the green and blue? Did you figure those out?
I put those as their single strand DNA.
Single strand DNA? Different ones, right? They're different? Or yeah, well, because obviously.
I'm almost thinking one is either one is RNA, but there's an
Yeah.
yeah. Like one is DNA, but the other is RNA, and the other is RNA, but both have an H, where an OH would be, so both DNA.
So they're single-stranded DNA, but they are different. Do we know like what type or what? No. And then,
If I did, I forgot it. Namely because I was trying to find waste. Namely, I was trying to get my ankle to recover. Multiple doctors.
are the green and blue molecules complementary to each other? No, not to each other, right? Not to each other.
They don't work complementary to each other.
But to the black, A, T, G, A, T, I don't know.
Are the green and blue complementary to a part of the black? A to T to C.
So wouldn't it be this?
T to A. There's no G to G. There's no G to T. C to T. So this is not complementary. G to A cannot be possible. G to A cannot be possible.
Kate Brown made it, and she is now in graduate school.
Because the
But transfer should be average.
for number 3, like
Name of the person.
Only the first first nucleotides are complementary.
Can I see what you did?
Let's see here. They're not completely complementary. Are the green and blue complementary to a part of the black? And
TAT.
yeah,
What is this?
I was trying to draw like a complementary.
I think I did the same. Let me see. Where am I? Oh, here. AGTTC. I did it backwards. I did it over here. T8.
Because otherwise I can't. So you actually have contact with 1,000 people times by years.
Yeah, I don't understand. So you tried that one, and then because when you turn this one, this should be A, right?
Yeah.
And then C should be.
It's on the form.
A, T, G, and C. So A to T and G to C. I think that's how it is.
Yes.
If I remember correctly.
Yeah, no, I think you got it right, but
Yeah. I wrote it my 222 notes, I believe. Yeah, A to T and G to C.
yeah. But then when he said that we only changed the T for A or the A for T, I forgot. That we only changed the T or
That's with RNA.
All right.
that's with RNA.
T becomes U.
So I emailed it directly.
Oh, T becomes U in
I can
RNA.
send it to you.
And RNA T.
Okay. Yeah. So how did you? Oh yeah, you're already in the next page. For the, how did you find out number 3? I mean number B and no. What, here, if they're complement. I know they're not to each other, but to the black one.
I just said they were because I'm pretty sure that's how PCR.
Yeah.
So I went and found the sections where they are
So you
reciprocal.
found. So
Oh, okay.
Oh.
Oh, I see it. Tot CA. I see it. And I did it, and
Oh,
I
okay.
just didn't find it
You
there.
have to make sure that you have the right ends, like which end is 3 prime,
All
which
right.
end
Oh,
is
did
3
you
prime.
get 5? Oh, I got it backwards. I got that as 3. Here, thank you.
Question.
Oh, thank you.
A question.
I got it backwards. Oh my god. Wait. Oops. Jesus, where's my catalytics? Oh. Not sure how to. Oh, that's a session there? Okay. What?
I talked to Charlie.
Single-stranded? No.
Yeah.
DNA.
I said single-stranded DNA now.
Okay, perfect.
Which carbon?
That's 1, 2, 3, 4, 5,
All right. What's on carbon 2?
and H.
Therefore, this is.
DNA.
Thank you. This is DNA.
Deox.
Are they complementary to each other?
No.
No, because if you flip them, you have to go in anti-parallel. This is 5 prime. This is 5 prime. So therefore, that has to be bound to there. So it's T to T. So that cannot work. So it's not complementary to each other. So DNA and non-complementary. What are these things called? These are called primers. What do primers do? They bind, right? So we had an enzyme called primase that starts DNA replication. Well, I put a primer down, now it gives me what end?
3 prime.
A 3' end. If I have a 3' end, what can I do to it? Build on it. Excellent. All right, so that's what we're going to do. You are in mastering, not running candidates. So line stuff on
A, C, A, T, G, and C. C does not go to T.
Oh, okay, there we go. Thank you.
This test tube to 95 Celsius, breaking hydrogen bonds. The tip upon just represents what the black molecule looks like. You then cooled this test tube to 60 degrees Celsius, temperature which the hydrogen bonds quickly reform.
Oh, this one here. Oh, you got it.
Yeah. The green, blue, and black molecules can come together, bind in 2 different ways. Based on
60 annealing.
yeah, it's in here.
So this one, scenario 2.
Based on which of these 2 possibilities is more likely to occur.
But it's split, so the top's the bottom. Yeah, just the top one is the bottom of strand. So you start to or no, I mean the top one, which is the bottom one. So scenario 2 is because scenario 2 is still pretty boring.
Yep.
So that's like why I
A binds 15, G binds 6.
I know.
What scenario do you think?
I was thinking number 2, because if you look at the PCR videos, they like show this
Yeah.
DNA splitting, and then these primers just go on to the split DNA and just shoot.
But based on these, it says based on these, right? Did you put scenario 2 or 1 or 2?
Yeah.
Yeah, because I saw the
guess it was
videos
a
and I was
PCR
like, it
videos
looks
that
like
we
the
watched.
second one.
This
But
is a PCR video
what
that
you-
we watched like 2 quarters ago.
yeah, but I got thrown off by the say C Model 1.
I wasn't going to come. I forgot that school started today. Wait, really? I know. I'm sorry. My parents' life is amazing. I had a house for like a whole week. Dude, they say it's so quiet. I'm kidding. They have colors, so it's not. Wait, what do we do with the colors? On question 6.
You're gonna start making the strings.
But I don't have colors.
He said to use your words.
It says to use the colors. This is extremely important.
Improvise.
Alright, I have a question for you guys.
Yeah.
You were in our heads.
Next question. Yeah.
I just know extremely important in my mind.
Would you like colored pen?
Do you have that? Dude, I love coloring. Wait, what costume?
That's I just wrote.
You're so boring. You're so boring. You don't know how to have fun.
I have so much fun. What are you talking about?
You don't.
Alright, 2 more seconds. Which one's more likely? Scenario 1 or scenario 2?
Scenario
2.
Scenario 2. And there are many more green and green and blue ones than there are
Black.
ones. Okay? Let's see. What does heating do? Breaks the hydrogen
Breaks the
bond, so it
hydrogen
allows this to unzip. In your cells, they don't heat to unzip. It uses what enzyme? Helicase. Excellent. So we don't use helicase here. Okay? When you get to this question, answer it.
One string of DNA for the fellow. So all these primers are going to stick for the DNA.
Information. So we just changed the letter? Do we just change all the letters or just one?
I think from the primer to.
Oh, yeah, I think
Wait,
that's.
in what color?
I only did.
Wait, no, but it is the bottom one. The bottom one is the top one. It's already switched. Wait, so. Wait, what? So are you saying we're doing A and Z? Like that's how it would. Look at the bottom one is the same. It's already
Just change the letter.
Ricky?
Oh, so start right there.
We just had a question with this one. We don't really know, like, it says right in all the sequences, but like, we're just confused by like that.
Yeah, so you find your primer is,
Oh, okay.
right, and you match the directionality, so this is.
So we just write, we just write the primer?
Primer and then the rest of the code,
Okay.
right? So you replicate it over and over and over again.
Wait, so that's in blue?
Oh, I guess, No, that's already over here.
So we just do the. Yeah.
And you're gonna do it again. So you're gonna teenager, yeah. I think, yeah.
And then it says continue.
How many strands of different
I had all of them. You had sequence strands and then you have another.
Wait, are they supposed to be going the same
Yeah, I put 4.
These were each like paired together. So then it has to be the opposite, right?
How many strands?
Wait. Let me just look.
Oh, yeah, the first one was 2.
Wait, I'm more happy. I thought you said this one's 5 through 3.
So the arrow is next to the 5.
The arrow doesn't make sense. I don't know.
That's right, yeah.
Like, I understand HDR, but this is
Yeah, I'm aware.
Or are they all the same thing? Why did you put two?
I just looked at the model and probably it's just showing that
What's the locations?
there's 2 other shoulders that
We did get it right.
So we did 2 new strands?
Are we supposed to find the length of the strands? But they just want me to
Like how many base pairs do they have? Okay.
Is it 2? There's 4.
So that's where we, right?
how do we know?
I think 2.
We don't know.
Okay, so it's just 2 and then this is.
I counted 20. Those are corals. Yeah, corals are
So question 9 is asking how does the enzyme know when to stop synthesizing? Is that asking like when it's talking to screen or when it can like when primers can know or the primers can no longer bind to the DNA?
No, it's the enzyme no longer, it runs off the end.
How does the
So
enzyme know
just,
when to stop synthesizing? When you find the stop or what? Huh? Right?
Like,
What was it?
UAGD.
Got that little table, Stop codon.
Isn't that what we did on the last question?
so what happens if I had it 32 times? You'd have to kill him. Is the stop
Not real sure. It was like little like plastic like so my dad he went I'm not that's what's fun. Okay, sorry I'm blanking, I'm so tired. But when I was like in like the 4th grade and he went to his like workplace, so like it was all his buddies. And so they found a pen on mine with plastic like sperms that like float like on the top of the pen. And he found it really funny, so he asked me to have it too. Because I was like obsessed with health back then. He loved it. And so I brought it to school and my teacher took it away from me. What is that word? And I found her using it the next day and I was so pissed off. I stole it. I used it. She said I was too young to be using something like that. I was like, really sad. But that teacher hated me. She made me wear jackets when I was like the senior. I was like, I'm never cold. But she forced me to wear jackets. I hated her. And she was like, draw my arms. Like what? Like you're not my mom, dude. Leave me alone. She also told me I only got this farm life and my friends actually like me, and I was like, I hope so. Like, why are you keeping with the fourth grader, dude? Do not put a friend like you guys. People there.
I'm not impressed with the fact that
There's no way.
12 years ago.
No, my dad was still. One time my teacher took yesterday, and he didn't come back in a year, but he promised, so my dad would share at him. It was the last day, 9th grade, and he was like, you have detention. My dad was like, I'm going to high school. You can't give me detention now.
25 minutes?
Yeah, I'm a lot like her.
This is weird.
No, I've worked on my anger, dude. I'm a lot better. Not 4th grade me. I'm just checking out.
Did you sign up for the sales job? It's like a hospital.
Okay, what? Oh, yeah. Yes.
How many, what did you get for 8A? I got 4.
4 and then 2.
Oh,
And then 25 and 2.
base pairs?
Well, we did. Was that base pairs? We just counted 25 for the shorties. 25.
Oh,
And nucleotides. Does that sound good? I mean, that's what we did over there. But, let me see. How many strands?
after the second cycle, because I was thinking 4.
Yeah, we did that. That is 4, and that's 2, and that's 25, and that's.
All right, so let's go through DNA replication for a second. So, arrow represents 3 prime, right? And so point of initiation is right there. And then in DNA replication, we have an enzyme that unzips. What enzyme is that? Helicase, excellent. And then we have what binds at the point of initiations? Primase, right? So a primer is going to stick down a sequence. And then in this case, we're going to be the leading strand because we're moving towards 3 prime. And that's going to be DNA polymerase, 3, right? And then in this direction, we're going to put things down. But we're going to have to do primers and then build backwards. And we call those what? Excellent, Okazaki fragments. That's DNA replication. What is the purpose of PCR? Replicate DNA. Okay? So instead of having proteins to initiate and start and stop, we're going to use heat. Heat's going to denature things, make things fall apart, break bonds, so forth. So I have strands, so that's 3'. This is 3', right? So they're complementary to each other. And if this is a sequence of, say, T, A, C, A, G, right? This must be
A, T, G, T, C.
excellent, okay? Now, if I want a primer I want to rebuild this side, I'm going to put a primer down that's going to be T, A, C, A, G, right? That is my primer. So this is going to be a 5' in, 3' in. Now I'm going to replicate that frame. If I want to go the opposite direction, I'm going to have to do the exact same down here with whatever it is. We're just going to say T, A, G, G, A, right? And then we're going to go in that direction. And that's going to give us our frame. So if I start with one strand of DNA, how many do I end up with after one replication? 2. After 2 replications, I end up with 4. There we go. 3 I end up with 8. 16, 32, 64. So each time it's going to use up more and more of the primers. Okay, so when we do PCR, I'm going to stick in a swab of the DNA, just like you guys did in 221. We're going to stick in our master mix, which is going to have a bunch of nucleic acids, nucleotides, and we're going to have a bunch of primers in there. Over time, what happens to the amount after each cycle?
Goes lower and lower.
Gets lower and lower. The amount left gets used up because we start making more and more strands. Okay, so the correct answer is 4, right? Yeah. Switching to the next question. 2, 1. I think everybody's in this.
Really think that they understand these sheets. Okay, we got that.
You got it, right? Yeah, isn't it 25? Oh, you're not counting this?
No, I'm counting this one.
Maybe you're right.
20?
Yeah, it's 20.
Oh. Yeah, because if we take out these 5, then we're at 20. Makes sense.
I just
Oh, like the word, oh, yeah.
I didn't see it.
Green blue primer, so that is.
Yeah, you're right. It is 20.
It is 20?
Yeah, because we don't count
Okay, 10
it.
seconds.
Oh, wait, I need to change my response. Okay.
Yay.
I didn't ignore.
All right. It is 20 nucleotides.
20.
Okay, so we answered 20.
That strands from cycle 1 are used.
Okay, next question. All right, talk to Sharon.
I picked black.
I said false strands.
Oh, so old.
Oh,
Oh, okay. Thanks.
well, let's see. Maybe
Yeah,
you're
let's
right.
see if we're right.
So what would happen if we only ever use the black strands as templates?
You could only ever create the same amount of.
You only ever create 2 more. You would never be doubling. So we're using all the strands. Because what's the purpose of PCR? To be able to physically see the DNA, to create bands on the gel. So we use all the strands.
Yep, you guys are right.
When you get to this one, answer it.
And then you said no, I meant, yeah, I know, but for A, you say E. That's what we said. For A. Yeah, I don't know.
If the second cycle was 4, why was the third cycle
So do you use strands? Do we get
Uh-huh. You can make more of them.
How do you go from 4 to 8?
Because you're using the templates and the ones you just made.
This one's not 200.
Okay, so here's your first two. The black ones is what was there originally. Then these, what you're finding there, those are your 2 new ones. And then, so we're going to have 4 total strands. So then I can make 4 new ones. No, you haven't made yours yet. These were there. This is what was made from the previous one.
and then the next page is the words.
What? Correct. And then you're going to make
And it just doubles each time.
correct.
Okay, that's don't ask. Don't ask.
I guess as long as we know that it doubles each time.
Oh,
Oh. Oh, that's right. I need to read. I need to read.
You're saying that you're only having two, so there's only two hours. When you split it, it's only 2 points. So
I'm just looking at it based on that.
oh yeah, then C is after it develops a little bit, and then and then this part
Blame me. Yes, correct.
So C would be like if I put it here, C would be generate these. And that.
Three is 8. 4 is 16. 5 would be 32. 6 would be 64.
Well. Wouldn't it just be 2 to the power of 10 and then 2 to the power of 9? 2 to the power of 10. Well, if it starts at 4, then 16. Right, so then 2 to 4, 2 to 10, yeah, 2 to 30. Yeah.
I was just wondering, I thought I those table.
I think sometimes, yeah, I think from another class.
Oh.
Wait,
Oh, that's right.
The gray one?
Okay, like 10 more seconds on this question. Okay, let's go through this one together. With the black, after 10,000 cycles, how many strands of black will you have?
2.
2. Doesn't matter, you're never doubling black. All right. Say after many cycles, how many of the B's do you get? you're doubling, right? So if you go from first cycle, you get 1 to 2 to 3, 4, 5, 6. It's not exponentially increasing. This one is, but you're producing most of these. D is the correct answer. D is our targeted sequence. So if we do our equation, it's like to the power minus 1 exponent. Okay? So D is our target. So if we look at bands on a gel, okay? So you have your gel, different bands. D is down here. It's the one it's gonna be the big, thick band, really clear C. The other ones might show up. They're gonna be longer, bigger fragments, so they're gonna be further up the gel, but they might show up. The original is there, but can you see it? No, it's too small. Right? There's not enough copies of it. So D is the correct answer.
We say there's a bug in the so it's exponential increase.
All right, next question.
What's the approximate number of first cycle number 30?
All right, what's the correct answer here?
2 to the 30th.
2 to the 30th, excellent. So let's talk about PCR for a minute. Remember, so in fall quarter we did PCR. You started with a small pellet of cells, and we did DNA isolation, okay? And then once you had that, you added your DNA to the master mix, and then we ran it through the thermocycler, which did the hot-cold cycle. Let's imagine you made your pellet, but it was too big, okay? So you had a big pellet of cells. You got isolated DNA. We isolated lots of DNA. Is that good or bad for PCR? What is the consequence of having a lot of DNA? Talk to share. What's the consequence of having a lot of DNA?
Would it be that you don't get a high concentration of the target strand?
That sounds logical.
That you're going for?
Yeah.
So ignore the question. I'm not asking a question. We want to use this diagram to illustrate this point. So what happens if you have a lot of black? You get a lot of the first scenario. Excellent. And we stuck in the master mix a certain number of primers and nucleotides. What happens if you have a lot of blacks? You use up in B and C because there's a lot of these. Will you get D? No, you'll get too few of D. So even if you run it 30 cycles, you've probably used up all of your master mix in cycle 5. So are you ever going to get that exponential increase of your target sequence? No. So several of you might have done everything correctly in the lab, all the dilutions, done everything, the isolation, but you started with the premise of too many cells because you said, oh, I can see that, that's excellent. We all wanted, how many times did I tell you cut that in the 10,000 smaller, right? How many cells did you really want? 1 or 2? And you were cutting these fragments, even the tiniest little dot of a pinhead was thousands of cells. So one of the consequences of that is failure. PCR fails. And every step was done correctly except for the very original. You stuck too much in. You never made it past C. So you never got your true bands. But then I said, send it out anyways. Why did we still send it out? Because those computers, the sequencers, are really good at finding stuff. And if they just find a strand, it will sequence it. So they were able to still find it even if you didn't get a band. But wait! Can you get a band and it still be incorrect? Could this, if this is just a large abundance of C, could that show up in a band in a gel? And not actually be D, right? So how professional labs do this? Okay, they make sure they have a very small amount of the original DNA. They do the correct number of cycles based on the size of that. They calculate how many cycles they want. And then in their gel, once they run the gel, they take a micropipette or a syringe and extract the band they are looking for. That's how to isolate and purify it. That's purification. So we did not do that in our lab because we just did a crude PCR analysis. But if you want something very specific, that's the way to do it. All right, what is the steps? Seconds. Right, Dr.
Denaturation, annealing, and extinction.
That's what I got. I might be wrong.
I think you're right.
Because we do denaturization, then we cool it down.
Annealing,
Yeah.
and then extinction.
Yeah. Okay. Yeah, because I'm recalling from that PCR video that we watched 2 quarters ago. Fall.
I watched a PCR video when I was doing the RQ0. You know, the quiz.
Oh wait, no, I put like that.
Scientists, the discovery of PCR was revolutionary because before, in order to do the thermal cycling, you would have to put in new enzymes every time after every cycle. But the discovery of DNA polymerase from this organism, which is lives in hot springs, is really important. For what reason? So talk to each other. Why do you think this is important?
Because it withstands the heat. That.
And that make also prediction.
And the others
what
don't.
that enzyme difference is.
What was the other question? Was there another question or just?
How hot is 70 degrees?
Party.
Really hot. Okay? Why is this one better?
Resists heat.
It doesn't denature. So what do you think is different about this enzyme on a structural level? What? Excellent. What R groups do you think are different?
That's why I don't know.
Sulfide?
Sulfide.
Disulfide.
They had disulfide bridge bonds. Instead of hydrogen bonding, it's using disulfide bridge bonds. It's able to withstand heat. Therefore, our last question was, we start with denaturing. We have everything fall apart. Then we cool it back down. This allows the primers to bind to the correct sequences. And then we have extension. Okay, so 4. Primers are binding to a specific sequence. If you want to look for a certain gene, you look for a specific sequence. You can order that primer from a store.
Amazon. Temu. Oops.
More seconds. You know, I am glad between every quarter at least one thing never changes. What is that? You don't listen to me. What did I just say about the primers?
Store.
They're made at a store. So why are we adding this? Not made by primates. We made the primers. We bought them at a store, okay? We did 3, okay?
Yes. First time I listened.
First time Mara listens. Oh my goodness. All right. Next question. Why do we use DNA primers instead of RNA primers like we would in DNA replications?
We don't have ligase to replace the nucleic acid.
Excellent. We don't have DNA polymerase 1 and ligase. So if we used RNA primers, okay, so we'll go back to our model again. These sections would fall off during denaturing, and your target would get smaller and smaller. And then, if this section falls off, will the next primer be able to bind to it? No. So that'd be a problem. So we have to use DNA primers. All right. So where do we get those DNA primers? A store.
Temu.
Literally, I order them. I put in the sequence I want. I press enter, pay $8.99, and they ship them to us. Okay, primers are very cheap. They're easily made. Yeah, you can get a primer for literally for any gene or any sequence in DNA. So, could this be an experiment you do? Yeah. What if you design a new lab, right? What if you decide, hey, I want to genotype something? That's an experiment that you guys can do. So then what do I need to have for you guys? Primers. We need to order primers. Now, primers take about a week to get, typically. But let's say you're doing that. Or we've had previous groups go around. One of my favorite ones, which was it was kind of funny, is they went around and they set up Petri dishes for 1 hour in every bathroom on campus. And they decided to see what's in the bathrooms, right? Which bathroom is the grossest bathroom? Because we're not going to do this. I'm not going to tell you because we might do that experiment. But then they decided, okay, so we figured out which is the grossest bathroom. And then what's the most common thing in that bathroom? So they PCR'd it. And they found out what was the most common thing in there. It's probably the bathroom you guys use most often.
What?
It's Shuckson's, women's Shuckson's bathroom here is the most gross.
No.
Women?
Tell me why.
Je ne sais pas.
Why is the women's bathroom here in Shuckson this floor the most dirty bathroom in all of campus? Take 10 seconds. Talk to each other.
And it's the bottom one, not the upstairs one. Because it's enclosed? I don't know.
All right, why? What's a hypothesis? A reasonable hypothesis?
No air?
No?
There's no air.
What?
The microbiology lab.
I like that, but why women's?
Is there more women in this building?
Pre-nursing. There are 5 to 1 women to men in this department. There are 5 women students for every male student in this department.
Oh.
That bathroom is used all the time. That's why it's the grossest. Should be the biggest bathroom. It really should.
Yeah, because
It should be
5
It's not,
times.
because there's 5 times the number of women. Okay? All right. With that said, what are we doing this week? Cure planning. You are spending hours this week planning your cure. You're thinking about things. You're communicating with me. All right? I don't want no surprise this weekend when I go and look and you're like, you have a whole doctrine of 10,000 pages of something you're going to do. I'm just kidding. No one's going to do that. But I really want a communication. We start this line. Okay?
So just via email or?
Email, text, I don't care. I want to have that conversation with you at some point this week.
Okay.
All right? Nice seeing everyone.